Hi, everyone. Thank you so much for joining us today. We have a very special guest speaker with us. We have Dr. Matthew Bernstein, who is a principal scientist in computational biology at Immunitas Therapeutics. He develops computational and statistical methods to analyze large-scale single-cell and spatial transcriptomics datasets to discover new immunotherapies for challenging cancers. And prior to joining Immunitas, he completed his Ph.D. in computer science from University of Wisconsin Madison, and his postdoctorate was from Morgridge Institute of Research. And broadly, he's interested in applications of machine learning to biomedical sciences. And this talk will cover the mathematics behind variational autoencoders and how variational autoencoders are being adapted and applied to the analysis of single-cell genomics data in order to find latent representations of the cell. So thank you so much, Dr. Matthew, for joining us today, and over to you.
Yeah, thank you so much. Share my screen here. Can everyone see my screen now?
Yeah.
Okay, great. Yeah, thank you so much. My name is Matt Bernstein. It's an honor to speak to you guys today. So today I'm going to give a primer on variational autoencoders and also discuss some of their applications in single-cell genomics, which is my primary field of interest. So a little bit about me. I'm a computational biologist. I work at Immunitas Therapeutics. We're a small biotech company in Waltham, Massachusetts, about 25 minutes outside of Boston. And we develop antibody-based therapies for immuno-oncology. In short, we develop drugs that seek to stimulate the immune system to fight off cancer. And I, in particular, develop computational methods for analyzing single-cell genomics data, which I'm going to talk about, with the goal of helping in the discovery of these new immunotherapies for treating cancer. I work at the intersection of biology and computer science, though my formal training is in computer science. I received my Ph.D. in computer science from the University of Wisconsin, Madison. And broadly, I'm interested in the application of machine learning to genomics, and I have particular interest in these kind of deep generative models and how they're being applied. And so my primary motivation for exploring and studying variational autoencoders, or VAEs, as I mentioned, comes from their burgeoning use in single-cell genomics. And I realize that most of the audience here likely has more of a computational background than a biology background. So let me give a little bit of a background into single-cell genomics and also some brief review and some basic molecular biology. So single-cell genomics, it's a sort of an umbrella term for a family of technologies that enable genome-wide so they enable one to produce genome-wide biomolecular measurements of individual cells. And the premier technology in this kind of family is called single-cell RNA sequencing, or scRNA-seq, where one sequences the RNA in each individual cell. And by doing so, one can measure the relative abundance of RNA coming from each gene. And so what do I mean by that? So now is a good time to give a very brief review into some basic molecular biology. So the cell is the basic building block of an organism. We're all composed of cells, trillions of cells. And within each of our cells, we have a copy of our genome, which is stored in our DNA. DNA is this long, double helical structured molecule that, this famous double helical structured molecule. And short sequences along the DNA are called genes. And those short sequences encode the explicit instructions for functional molecules that perform the biochemistry within the cell. Those are predominantly proteins. And so when a cell wants to create a protein or express a gene or express the protein, it does the following. So first, it copies that gene, that sequence of DNA, from DNA to an intermediate information storing molecule called RNA. This RNA molecule is then free to float away from the DNA, where it meets up with the cell's machinery that then translates that RNA into a protein. And these RNA molecules are often referred to as transcripts. And so in single-cell RNA sequencing, we sequence this RNA in order to measure how much RNA is coming from each gene relative to all the other genes in the genome. So the end result is this high-dimensional vector, where each element of the vector tells you how much RNA is coming from each gene, relatively speaking. And there are about 20,000 protein coding genes in the human genome. So this vector is usually on the order of about 20,000 or so genes long. So each cell then is represented mathematically as this high dimensional vector. And it's common for a single cell study to produce thousands to, in some cases, even millions of cells at a time. And this is enabling high resolution exploration of biological systems at an unprecedented scale. So here's sort of an illustrative example of a data set published by Yao et al. Of over 7 million cells from the mouse brain. So here on the right, I'm showing a t-SNE plot where each dot represents a cell, and these are two-dimensional embeddings of these, what are 20,000 or so dimensional vectors that I just described. And there are about over 7 million, I believe, in this study, and they're colored here in this plot according to what cell type they are in the brain. And so one key thing to know about this kind of data is that these vectors lie on a lower dimensional subspace. And the reason for that is that genes are not expressed randomly, but rather they're expressed in a highly coordinated fashion within the cell. And the result of this high coordination is that these vectors lie in a lower dimensional latent space, so to speak. And that's depicted on the right here in sort of a cartoon fashion here as sort of a schematic, where this three-dimensional space represents this 20,000 or so dimensional ambient space. And then this Swiss roll is just kind of a cartoon representation of this lower dimensional subspace in which biological cells reside. And as computational biologists who look at this kind of data, we're often interested in understanding the properties of this latent space. For example, some regions of this space may be inhabited by healthy cells, whereas other regions of this space may be inhabited by diseased cells, and then cells that lie somewhere in between along this subspace may represent cells undergoing some kind of disease progression, sort of a hypothetical example. And so there's a lot of research in computational biology in developing methods that will extract this latent space, that is, find these latent representations of cells from the high dimensional data, from the high dimensional raw data. And variational autoencoders are one such tool for doing this. So VAEs are used for learning these latent representations of cells. And these latent representations can be used for a number of important downstream analysis tasks, such as visualization, separating technical effects from biological signal, imputing missing measurements. I'll talk about how single cell data is actually quite sparse. And so these latent representations enable one to develop a sort of Swiss Army knife of tools depicted here on the right. And this analogy comes from a commentary by Wei and Green. And there's a popular Python package called SCVI tools that implements variational autoencoders for doing single cell data analysis. And so from a personal level, in my day-to-day, I analyze single cell data, and I sometimes use these tools. And so I wanted to gain a deeper understanding into the mathematical foundations behind variational autoencoders so that I could really understand, how they work, what tasks they may be better for or less suited for, and really and also understand when the methods are not working. So I went about kind of just teaching myself this material. I read the original papers, although that's always a little challenging. They're kind of terse. So I watched YouTube videos and blogs. I read blogs. And so today I'm going to talk about two things. So first, I will provide an in-depth tutorial into the mathematical foundations behind variational autoencoders as I understand it. And then I'm going to come back to the biology and talk a little bit more in detail how variational autoencoders are adapted for analysis of single cell genomics data. So there's kind of two target audience members that I hope get something out of the talk. So one is somebody who would like a kind of deeper understanding into variational autoencoders and the mathematics behind them, and somebody who's interested in just kind of a high level description of how machine learning is applied in a certain area of computational biology. So starting with the tutorial into the mathematical foundations behind variational autoencoders. So what are variational autoencoders? They are a family of deep generative models, first developed by Kingma and Welling in 2013, and they enable two essential tasks. So first, they perform dimensionality reduction of high dimensional data. As I said, we're trying to find these low dimensional latent representations of cells. That's what we're going to use these variational autoencoders for. And two, they enable for modeling and sampling from complex distributions. So they are generative models. One can generate samples from the distributions that they encode. And to do these two tasks, variational autoencoders combine the conceptual frameworks of Bayesian inference and deep neural networks. And there are two complementary perspectives from which one can understand variational autoencoders. The first is more fundamental, more foundational, and that is that variational autoencoders at their heart describe a probabilistic latent variable model. That is, they describe a joint distribution P of X and Z, where X is our observed high-dimensional data points, and then Z are the latent, unobserved, lower dimensional representations of these observed high-dimensional data points. And as I'll talk about, from the specification of this joint distribution, coupled with the method that we're going to use to do inference on this distribution, emerges a sort of autoencoding neural network. And as a brief review, an autoencoding neural network, is this neural network architecture for which you give it some high-dimensional data, X, and the goal of the neural network is simply to reconstruct X. So X prime here is the output, and the model seeks to create X prime so that it is as close to X as possible. And this might seem trivial, except for the fact that we force the model to first pass X into this lower dimensional representation Z. So it has to squeeze X into this lower dimensional vector Z before it reconstructs the input. So by passing it through this information bottleneck, this so-called information bottleneck, the model is forced to extract the essential information from the input that it will need to then reconstruct it in the end. And I'll talk about how this emerges from the specification of this joint distribution and the inference algorithm. So starting with this first perspective, variational autoencoders are latent variable models. As I mentioned, a latent variable model is just a joint distribution over two random variables, X and Z. X is observed, so we have that, that's our data. And then Z is unobserved. So we don't have access to it, but we posit that it exists. And then theta are the model parameters. And then in variational autoencoders in particular, X, as I mentioned, are high dimensional data points, and then Z are their corresponding lower dimensional data points. And so variational autoencoders define this joint distribution in a very particular way, where one can sample a high dimensional data point X and a lower dimensional data point Z through the following generative process. So first, we sample Z from a standard normal distribution. That's how we get Z. Then we pass Z through a function, f sub theta, and this is where the parameters theta come into play, to form this higher dimensional vector psi. Then psi is used to parameterize some distribution D, from which we then sample X. And in practice, f sub theta is usually a neural network. So this encodes some complicated nonlinear function between the lower dimensional space in which Z resides to the higher dimensional space in which X resides. So depicted schematically in a slightly different way, we start with this normal distribution, usually a standard normal distribution from which we sample Z. We pass Z through this neural network to get this high dimensional vector psi. And then we use psi to parameterize D, from which we then sample X. Depicted a third way, here I'm showing a sampling of two latent representations Z from P of Z, which is just that standard normal distribution. They're mapped to the high dimensional data space where they parameterize these conditional distributions of P of X given each Z, from which we then sample each X. And the thing to note is that f sub theta enables us to define a very complex distribution. So when we look at the marginal distribution of X for getting the Z's, that can be quite complex. And that complexity comes from this function f sub theta, this neural network. So that's kind of the gist of the generative model. It's essentially just encoding some complicated distribution where that complexity comes from the neural network. And so there are two kinds of inference tasks that we may be interested in doing on this model. So as I mentioned, given some high dimensional data point X, we're interested in its low dimensional latent representation Z. And we can get at z by examining the posterior distribution of z given x. And this will enable us to understand the most probable values for this latent representation. And naively, one can apply Bayes' theorem, although as I'll talk about, that's intractable to compute exactly. And then a second inference task is to find the parameters theta that fit the model to the data. That is, that make the data most likely under the model. So we seek a maximum likelihood estimate of theta. And for that, we just want to find theta that maximizes the likelihood here of x. Unfortunately, as is the case in many instances where we're trying to do inference on non-trivial statistical models, both of these inference tasks are intractable to compute directly. So if we try to use Bayes' theorem to get at this posterior, we have to deal with this normalizing factor in the denominator. We don't have this marginal distribution over x, so we have to marginalize over all possible z's because we do have access to the joint distribution, but this integral is usually hard to calculate, so we can't really do this. And then similarly, unfortunately, we also have to calculate this integral when we look at the likelihood function p sub theta of x. And so both of these inference tasks are intractable to at least compute directly, so we're going to need some way to get at these solutions in an approximate manner. And in variational autoencoders, this is done by applying variational inference. Variational inference is a general framework for doing Bayesian inference, for computing approximate posterior distributions. And the way that it works is, instead of calculating the posterior directly, p of z given x directly, we will instead estimate it using a surrogate distribution, q of z given x, where we define this distribution, q of z given x, so we know what it is, but we make it parameterizable by these parameters phi that we can then tune so that q of z of x is as close to p of z given x as possible. So that's going to be our goal. Our goal will be to find the parameters phi that make q of z given x as close to p theta of z given x as possible. And this will actually be possible to do even without knowing the true posterior. And so what do I mean by as close to as possible? In variational inference, we use KL divergence from p of z given x to q of z given x. And KL divergence, it comes from information theory. One can think of it as a sort of similarity between distributions, although it is asymmetric. So one can think of it as a divergence from this distribution, p of z given x, the true posterior, to this approximate posterior. So that is going to be our task. Our task is going to be to minimize this KL divergence from the true posterior to our approximate posterior, and to find the parameters phi that minimize this KL divergence. And one can write this KL divergence in the following form shown down here, where here you can see the log likelihood function, which doesn't actually involve phi. And then this second term on the right, which does involve phi. And notice that because this term here doesn't involve phi, we can kind of forget about it when we want to minimize the KL divergence. And because of this negative sign, to minimize the KL divergence, all we need to do is maximize this term on the right. This term on the right is called the evidence lower bound, or the elbow. So this is our new objective now. Our new objective is to maximize the elbow with respect to phi. And that is given by this term. It's a little ugly looking, at least at first, but something to note about it is that nowhere in this term do we see the true posterior of p of z given x. So this is kind of a trick, I guess, so to speak, where we've come up with this objective function such that if we maximize it, we're going to find this approximate distribution, q of z given x, that is going to be similar to p of z given x, even though we don't actually know what p of z given x is, because you can see it doesn't appear anywhere in the elbow. So what about theta? What about the second inference task of fitting the model to the data? And it turns out that we can use the elbow for finding theta as well. And we do that by noticing that the elbow is a lower bound for the log likelihood function. So here's the KL divergence term again, and we can rearrange it so that the log likelihood is now on the left. And we note that the KL divergence is always non-negative. And so if we know that, so we do know that, and because of that fact, we see that the log likelihood function is always going to be greater than the elbow. And as a side note, another term for the log likelihood function is called the evidence. And the term on the right is a lower bound for the evidence, hence the term evidence lower bound. And we note that if we maximize the elbow in terms of theta, then we will be maximizing a lower bound of the data log likelihood. And if that lower bound is tight, so here the elbow is in red and the log likelihood is in blue, if that lower bound is tight, then in an ideal situation, maximizing the elbow with respect to theta now, forgetting about phi, will give us a value for theta that is close to the maximum likelihood estimate for theta. So our task now is going to be to maximize the elbow, not just with respect to phi, but also with respect to theta. So this is the final objective. We can and we can do these two inference tasks simultaneously by, that is, finding an approximate posterior distribution and finding a maximum likelihood estimate by maximizing the elbow with respect to both of these sets of parameters. So that is essentially what we're going to do. But we haven't really gotten to where the autoencoder comes from. And that's because we haven't actually defined what Q is. So what is Q? How do we actually define Q? So in variational autoencoders, Q is defined in the following way. And remember, this is the distribution that we're using to approximate the true posterior distribution. And it's defined via the following generative process. So we start with X because we're conditioning on X. So we start with X. And we pass X through two neural networks. The first will give us this value mu, and the second will give us so that's a mean, and the second will give us a variance sigma squared. In practice, usually it's log sigma squared because so then it can be negative, but we're really just trying to get a variance. This mean and variance then parameterize this normal distribution from which we then sample Z. And this generative process will produce these latent, posterior samples. And we can adjust this distribution through these two neural networks. So depicted schematically, again, we start with X. We pass it through this neural network, or these two neural networks. It can be one or it can be two. It's kind of general. To get these lower dimensional vectors, mu and sigma squared, and these are our mean and variances, and they're lower dimensional than X, because they parameterize this normal distribution from which we then sample Z. And so here now we can see that we get an autoencoder. An autoencoder sort of emerges from our definition of the generative model and from our approximate posteriors, where the variational posterior, that approximate posterior, is sort of an encoder because we give it an X and it will produce this latent embedding Z. And if we start with Z and pass it and then generate a new sample X from our generative model, we get back X. So here's just an example of a result of a small variational autoencoder that I trained when I was trying to study this material. So this is an example using MNIST. So these are images of handwritten digits, just people writing numbers. So this is a handwritten 7. And here I'm depicting the process of sampling this latent representation of this 7 from Q of Z given X. And this model has already been trained. And then we then sample X from the generative model conditioned on Z. And then we get back something that looks like a 7. So we can see that this is doing autoencoding. I think that's kind of neat. So we start from a statistical model, define a certain inference algorithm, an inference model, and then we get out this autoencoder. So how do we then fit this model? And I think the devil's in the details. So now I want to talk about how we actually go about fitting this model in practice. So let's examine the loss function on a data set. So now I'm rewriting the elbow. So in a scenario where we assume that we have n samples in our data set, rather than writing it in sort of a general way where X is our observed data and Z is our latent data, here now we have n discrete points. And the elbow then would just become a sum of all the elbow terms for each individual data point. And one thing that we want to do, so this is our objective function, and because they involve neural networks, we kind of turn to gradient descent, or in this case, gradient ascent, because we're maximizing the elbow. But the problem is that we can't really apply gradient ascent because we have to deal with this expectation, which is defined by this integral. And this integral this expectation is over the distribution that involves the parameters that we're trying to maximize. So we can't really maximize this function, at least directly with gradient ascent. So we're going to need to come up with some way to get around this integral. And so in the next few slides, I'm going to talk about the method that Kingma and Welling in their original paper presented for circumventing this integral and dealing with it. And that is specifically something called the reparameterization trick. And so when I first learned this material, it wasn't quite clear like how this worked or where this got us. And so I'm just going to first present what the reparameterization means. And then in the next few slides, I'll talk about what it gets us, like how it enables us to get around that integral. So the goal is going to be to find a function g sub phi of some variable epsilon and x such that if epsilon is a random variable that is sampled from a standard normal distribution, and then we pass epsilon along with x into this function g, we get an effective sample z from q of z given x. So here we're essentially reformulating the generative process for sampling from q of z given x. So recall that beforehand we defined that generative process here. We're going to reformulate this generative process not to look like this, but to instead look like this, where g sub phi is going to be some function that we're going to have to define, but the end goal is going to be that we're going to get a sample z from this distribution, q of z given x. So this is depicted down here where we sample epsilon from a standard normal distribution, so it's just Gaussian noise. And then when we map epsilon along with x through g, we get a sample from q of z given x. And q of z given x can be a pretty complicated looking distribution because it's parameterized by these neural networks. And so what does that actually get us? So why are we doing this? So first, let's just write the original formulation of the elbow shown here. And this is what I showed a couple slides ago, is just the elbow. And then we can apply the reparameterization. And so if we apply the reparameterization, then we just replace all of the z's with the output of this function, g of epsilon and x, where now the expectation is over epsilon, not over z. So one thing to note is that already, so two things to note. The first is that these two forms of the elbow are mathematically equivalent. The second is that the expectation no longer involves a distribution parameterized by parameters that we're trying to optimize. So that's the first thing to notice. Then the next thing is that we can approximate this integral now, this expectation, by sampling values from just a standard normal distribution and then just replacing this expectation with an average. So we can get an approximation of this elbow through doing a Monte Carlo approximation of the integral represented by this expectation. So this is essentially the reparameterization trick, where we've gotten rid of the integral by approximating the integral. And moreover, this approximate elbow now doesn't involve an integral, but it's also can be differentiated with respect to phi and theta, so long as g sub phi is differentiable and p sub theta is differentiable. So that's essentially the reparameterization trick, where we can take we'll see we can take gradients of. But so far, we haven't actually defined g sub phi, so let's get to how is this actually defined? Well, this is actually quite easy in our case, based on how we've defined q sub phi of z given x. Because q sub phi of z given x is just a normal distribution, whose mean and variance come from these two neural networks, it turns out that z can be sampled by simply sampling epsilon from the standard normal distribution, scaling epsilon by the standard deviation, which we get from the neural network, and then shifting it by the mean, which we get from the other neural network. And then this becomes our function, g. So that's pretty much the gist of, that's kind of the big ideas in variational autoencoders is we get this autoencoder from how we specify this joint distribution and as well as this approximate posterior, and then we maximize the elbow by approximating the expectation using these Monte Carlo samples and the reparameterization trick. And note that we're calculating an approximation of the elbow, not the exact elbow. So if we take gradients with respect to this approximation, we're getting approximate gradients. And those approximate gradients actually have an expectation whose gradient is the gradient of the elbow. Thus, we can view this as stochastic gradient ascent. And in the original paper by Kingma and Welling, they actually went a step further than what I've just described, and they proposed a way to reduce the variance of these stochastic gradients further. So we always want to reduce the variance of our stochastic gradients, because if these gradients have a high variance, then although in expectation we might get to a local optimum of our objective function of our elbow, in practice we might not, because the gradient is causing us to kind of bounce all over the place. So we want to reduce the variance of these gradients, and we can do that with the following way. So let's take a step back, and here we rewrite the elbow. This is just the elbow again shown here. And with some derivations, one can derive the following form, where here we have this KL divergence term, and then the second term only has this expectation. And this reduces the variance because one notes that both of these distributions are just two normal distributions. Q of z given x is a normal distribution. And then P of z, remember, that's just a standard normal distribution. That's how we sample z in the original generative model. And the KL divergence between two normal distributions has an analytical form. In our case, that form looks like this, which looks a little bit ugly, but the key thing to note is that there's no integration. You can calculate it exactly. And this reduces the variance because we've sort of factored out some portion of the elbow to this closed form expression. And the expectation now that we're going to have to approximate using our Monte Carlo approximation is only in the second term. So we've kind of relegated the stochasticity of our estimate to this second term, and we've factored out some of it into this analytical term. So here's the final objective kind of written out in its whole form. I like to write it out just to kind of see it all in one place, where on the right here is this KL divergence term, and on the left here is this KL divergence term, and on the right is this expectation, which is called the reconstruction term, and I'll talk about why that is. And we approximate it with our Monte Carlo samples. And to note, to calculate the elbow over the full data set, one has to average over all the samples in the data set. One can approximate this further with mini batches, which adds another layer of stochasticity, but it's still stochastic gradient ascent. So why is this called the reconstruction term, and what is this KL divergence term doing? Turns out we can actually gain even more insight into this objective function. And to do that, let's make this model a little bit more specific. So recall this joint distribution of P of x and z. I had made it a little bit general, where psi was just some parameters and d was some distribution. Let's make it more specific, where psi is now a mean mu, and d is a normal distribution parameterized by mu. And we'll assume that the variance is some fixed hyperparameter. With this generative model, the elbow has the following form, shown here, where we see this mean squared error term. So what this is showing is that to maximize the elbow, the model will seek to minimize the difference between the original data x and the output of our neural network f sub theta. And this you can think of as the decoder. So this is not unlike the standard autoencoder, where we're trying to minimize the reconstruction of the output. So that's why this is called the reconstruction term. And this is just a constant, so one doesn't have to really pay much attention to that. And then the second term here, let's talk about that a little bit. So this is a KL divergence between P of z and our Q of z given x, our encoder, so-called encoder. And we see that what this is going to try to do is this loss function is going to end up doing is it's going to force the encoder to generate these latent representations Z that are distributed similarly to P of Z. And P of Z, recall, is just a standard normal distribution. So this is, so, and right, so let me just demonstrate what that looks like. So this again is the model trained on MNIST. Here I'm plotting the first 2 dimensions of the latent embedding in a test set where each point is a single digit and they're colored by what digit they are. And you can see that the first 2 dimensions are kind of spherical. They look like a standard normal distribution. If one then just plots the empirical distribution of the first dimension of the latent embedding, shown here in blue, you can see that the model kind of got that distribution to look like a standard normal distribution. And that's shown in orange as just the density function for the standard normal. So this gives us a little insight into what this loss function is doing. It's trying to accomplish 2 tasks simultaneously. One, it's trying to minimize the reconstruction, so it's doing autoencoding, but it's also trying to make it so that these latent representations are distributed according to a standard normal distribution. So that's kind of elegant. Like you can kind of see these 2 competing tasks, or not really competing, but 2 tasks that it's trying to solve simultaneously. And moreover, it turns out that the implementation of a variational autoencoder in many cases is not so different from that of a standard autoencoder. I found this kind of surprising because the math and statistics behind variational autoencoders is a bit more involved than that of a standard autoencoder. But the end result is oftentimes not so different, where in the case of a standard autoencoder, one computes the reconstruction error and then back propagates that error through the neural network to update the parameters. In the case of a variational autoencoder, we also compute the loss, which in this case is not just a reconstruction term, but also this KL divergence term to get these latent variables to look like a standard normal distribution. But nonetheless, we have a loss function that we then propagate backwards through the neural network. One thing to note is that when we actually run the neural network forward, we do have to interject this stochastic epsilon, and that's sampled from the standard normal distribution. And that's because we need to approximate the expectation in that elbow. And this is sort of injected from the side. One thing that a variational autoencoder can do that a standard autoencoder cannot do is you can generate samples from it. And one does that by simply running the generative model, sampling from that generative model. So once this neural network f sub theta is fit to generate a sample, one just first samples a latent vector z from the standard normal. Then one passes it through that neural network, then samples x. So here are some digits from that little toy variational autoencoder that I trained. And these were generated by the model. So there are 3, as far as I can understand, there are 3 main advantages to variational autoencoders over standard autoencoders. So the first, as I just mentioned, is the ability to generate new samples. So variational autoencoders are generative models. Second, they enable finer grained control over the structure of the latent space. So in the standard or canonical variational autoencoder, we enforce the latent embeddings, those z vectors, to be distributed according to a standard normal distribution. And this is not something that we can do in a traditional autoencoder where the model is just creating these embeddings that are kind of arbitrary depending on what the model needs to do to minimize the reconstruction loss. And then third and finally, by defining the decoder and the encoder as probability distributions, variational autoencoders are somewhat modular in that we can substitute new distributions into this framework to model different types of data. And this is one of the key aspects of variational autoencoders that make them suitable for single-cell genomics analysis. So now let's come back to single-cell data, applying variational autoencoders to single-cell RNA sequencing data. Right, so now that we've kind of gone through some of the math behind variational autoencoders, let's talk about how they're being adapted to single-cell data. Kind of switching gears, recall from the beginning, our goal is to identify these latent representations of cells. where our input are going to be these gene expression profiles, these vectors of these gene expression measurements. Here's like a whole data set shown here as a gene expression matrix, that where the rows are genes and the columns are cells. We pass it through a variational autoencoder, and we get these latent representations for these cells. And so to understand how variational autoencoders are applied to single-cell RNA sequencing data, I think it helps to understand some very basic ideas behind how single-cell RNA sequencing works. And I don't want to go too in-depth. I just want to get to some kind of core messages. But I'm kind of going to walk through at a really high level how single-cell RNA sequencing works. Recall again that when a gene is expressed, that gene, that is that sequence in the DNA, is copied to an RNA molecule. That RNA molecule, that transcript, is then translated into a protein. And our goal is to sequence these RNA molecules. The way that that works is shown here on the right, where we extract these transcripts from our cells. We perform this process, this physical chemistry, where we convert these transcripts, these RNA molecules, into a cDNA library that's fed to a next generation sequencing machine. A sequencing machine, it just sequences DNA. So we're going to leverage next generation sequencing technology. That generates these sequencing reads, which are aligned to the genome. And then one simply counts the number of sequencing reads that align to each gene. And based on the relative number of sequencing genes that align to each gene, one can gain an estimate of how expressed each gene is relative to the other genes. And that's kind of complicated, and I apologize, it's a little bit of an information dump, but the core, I think the really core message is that the output of RNA sequencing, that expression profile, that expression vector, is discrete counts. So these are discrete counts, they're not continuous values. Moreover, these discrete counts have specific statistical properties. So it's been found that these RNA sequencing reads, or UMI counts, can be modeled pretty well by a negative binomial distribution. And in the case of single-cell RNA sequencing, as opposed to bulk RNA sequencing, these gene expression profiles are quite sparse. So we don't actually measure most of the genes in each cell. It's very sparse. The technology is just not quite sensitive enough to get a sensitive measure of expression for all the genes in the genome per cell. And so to model this sparsity, some tools use what's called a zero inflated negative binomial distribution, where one assumes that there's some probability of not seeing that gene, so that it's just zero, or there's some probability of seeing the gene, in which case it's distributed according to a negative binomial distribution. It's sort of a mixture. It's a mixture model, essentially. And so in 2018, Lopez et al. Had the idea to use variational autoencoders to model single-cell RNA sequencing data, and to use this zero inflated negative binomial distribution in the generative model that is part of the decoder. So in this case, this D distribution here would be this zero inflated negative binomial distribution. And it's a little bit more complicated. So here's the full graphical model from their tool in plate notation. There's, again, so this in the end describes a zero inflated negative binomial distribution, but they also enable one to condition on different batches of cells or different patients from which the cells come from. They also enable modeling certain aspects of the technical RNA sequencing protocol, such as the library size. That's the number of reads one generates in the sequencing experiment. The key thing to keep in mind is that, the key thing that I want to point out, I should say, is that because variational autoencoders are sort of modular, we can model different kinds of data with different kinds of distributions. And here the authors created a distribution that models single cell data, and they kind of drop it into this variational autoencoder framework. And then by running that framework, we can get these latent representations of cells. And what can we do with these latent representations of cells? Well, For example, we can explore that latent space that I described at the beginning. So here's a nice illustrative example of such a study where the authors, Don et al., used this tool, this variational autoencoder called SCVI, to identify these latent representations of cells. Specifically, they looked at immune cells from the blood of patients with COVID-19 and healthy samples. And so here are these UMAP plots, these two-dimensional embeddings of these latent representations that came out of the model, where the pink cells come from COVID-19 patients, and the blue cells come from the healthy controls. In the middle here, they're just colored by what cell type they were deemed to be. And then on the right, they're colored by how much each cell is expressing an interferon signature, which is essentially a gene expression program that's activated when a cell is fighting off an infection. So this is just an illustrative example of how of a real data set where a group applied these models and were exploring the latent representations that these models extracted. And you can find these groups of cells in the latent space that come from diseased samples. You can kind of better understand the disease process. Variational autoencoders are used for other tasks as well. For example, they can be used for imputing missing measurements. I talked about how single-cell RNA sequencing data is sparse, so we don't get a measurement for all the genes. In fact, quite a number. In fact, the majority we don't actually measure. And we can use variational autoencoders to impute missing values. And we do that by passing in the raw data, the raw expression profile, which has these missing values through the model. And then when we get the output, the model sort of fills in these values and it sort of imputes them as to what the model thinks that they should be had they been observed. And this is very similar to how traditional autoencoders are used for example, denoising images, where one gives autoencoder some corrupt or noisy image. By passing it through the autoencoder, it denoises that image. Another use case for these tools is in removing non-interesting sources of variation. So oftentimes, cells from different patients will have a lot of variability that is inherent to the patient, or they can be due to technical effects. Like if that tissue sample was handled differently, the cells may be responding to the conditions at that time. So you get these technical effects, sometimes called batch effects, that are not necessarily of interest to the scientists doing the study. So here I'm showing a UMAP plot of cells coming from different donors. And if one looks closely at the plot, you can see that some cells, it seems that they cluster by the patient that they come from. So one always has to be careful when interpreting UMAP plots, but this plot would suggest that potentially there are some donor-specific effects in these data that we may want to correct for. And recall I mentioned that this model has this variable that tells the model which batch or which patient each cell comes from. And that enables the variational autoencoder to learn these latent embeddings that condition on this patient. And the end effect is that these latent representations become mixed. So it sort of mixes the cells together coming from these different patients or these different batches. So here on the right is a UMAP plot, again, of the latent representations coming from the model. And at least this UMAP plot would suggest that the variational autoencoder is doing some job at removing the patient-specific effects. And then lastly, I thought this was a creative use for variational autoencoders. So this is from a study published by Svensson, Beltram, and Pachter in 2019 as a preprint, where they explored using variational autoencoders to identify the optimal parameters to use for a given single-cell sequencing study. So going back very quickly, before I explain this figure, going back very quickly to single-cell, this RNA sequencing protocol, there One thing to note is that sequencing is actually quite expensive. And so one usually has a sequencing budget from which one gets a certain number of sequencing reads, some amount of data in the output. And if one provides as input too many cells, then one will get out a very sparse reading of each cell. So there's always a trade-off, there's always a question of how many cells to sequence. If you sequence fewer, you get more information about each cell, but you get fewer cells. So in this study, the authors wanted to see how different numbers of cells and different numbers of sequencing reads, how much sequencing to do, what is sort of the optimal level. And they did that by taking a data set, a single cell RNA sequencing data set, and downsampling that data set to some number of cells and some number of sequencing reads, some lower amount of data. And then they trained a number of different variational autoencoders on these different downsampled data sets. They then applied these different trained models to a held out data set and examined the reconstruction error. And essentially what they found was at a certain point, there's sort of a threshold by which if you have too few cells or too few reads, then the model does not do a good job at reconstructing a test data set, which indicates that you don't really have enough information to capture the statistical properties of the cell population that you're studying. So I thought this was just a creative use for these variational autoencoders, sort of outside direct single cell data analysis. And there are other use cases as well that I'm not going to go into today. That's all I wanted to talk about. In summary, variational autoencoders are a family of probabilistic generative models. They perform dimensionality reduction. They also enable modeling complex distributions. And variational autoencoders, they are these autoencoding neural networks that arise from the definition of a particular probabilistic latent variable model coupled with the inference algorithm. And what comes out is this autoencoding model. And due to their foundation in probabilistic modeling, they can be adapted to model a wide variety of data. And I showed an example of how they're being adapted to model these discrete counts that come from single cell RNA sequencing. And we're seeing a burgeoning use in single cell genomics, and I find it really interesting. I'm a user of these tools from time to time, and yeah, I think it's a really interesting application of machine learning to computational biology. And so with that, I just want to thank you all for listening, and in the remaining time, I'm happy to take any questions. Thank you so much.
Thank you for the talk. We have a question in chat that how many cells do you tackle at a single time?
Yeah, so I'll talk about what's usually produced in a given study. In a given study, it's on the order of, on the low side would be 10,000, and on the high side would be hundreds of thousands to, in really high cases, millions of cells. I believe that a single run, a single sample would have around 10,000 cells in most platforms, but usually in a study, one does multiple batches of cells, and so one gets tens of thousands of cells.
Right. Also, I wanted to ask that do you have any intuition on what should be like the property of a good encoder in with VAE, like if we talk about different architecture of CNNs or vision transformer, what is like something that works really well with VAE?
Yeah, so I guess in computer vision, one can get at that more directly by just looking at the reconstructed images. Visually, one can get a sense of how the model is doing. It's harder to do that in single cell data where one just has these large gene expression profiles. So one can look at the loss function. One can also hold out test data set and examine the reconstruction loss on those cells. And so SCVI has that capability to do that. I would say it's probably in the same vein as most machine learning, where you need some validation set and some function that tells you how good the model is doing on that validation set.
Right. We have another question in the chat. Can you comment on the explainability of VAE?
Yeah, so the explainability of VAE will probably depend on the particular network architecture that you're employing in these neural networks. So if you use like a fully connected neural network or some other architecture, that may be less explainable than an architecture that models some real world thing. So in biology, for example, there's many methods that use neural networks where the architecture follows some biological framework, such as gene pathways or gene sets. In that case, you automatically get sort of an explainable model at the end. But if you use a model that's not explainable, at least out of the box, then I think you'd have to turn to the explainability literature and try to interrogate your model and understand, what it's doing. That I'm not an expert in that field.
Another question in the chat: how did you move from CS into computational biology?
Yeah, so I'd always been interested in biology. I was originally a pre-med major, like my first semester in college, but I don't really handle that kind of thing very well, like blood and stuff. So I ended up switching to computer science, but I always had that kind of interest in biology. And then in grad school, I wanted to do machine learning. And then a lot of the research in grad school in labs and at the University of Wisconsin were geared towards applications in biomedical areas. So that was kind of a good opportunity to combine kind of my background interest in biology with computer science. And I joined a bioinformatics lab in grad school.
Another question from the chat: what is the current scope of diffusion models for generative processes you mentioned in genomics as compared to VAE?
Yeah, so variational autoencoders are used a lot in the single cell genomics. I have not really seen diffusion models used as much, as far as I know. But diffusion models are being employed quite a lot in a different area of computational biology, and that's in structural biology. So producing proteins, producing protein structures that have some specific properties, that's a huge area of research where they're using diffusion models to produce these protein structures.
Right. Another question: how have you tried other generative models?
So I kind of so in my day-to-day work, variational autoencoders are the most used because that's what's predominantly used in single cell genomics. As far as generative deep learning models go, in my day-to-day work, not so much, but I'm kind of on my side. I'm keeping tabs on diffusion models and their applications in protein structure science. That I'm finding really interesting, though in my day-to-day work, I don't use those models.
Another question: I am currently using CNN for phenotype prediction using multi-omics, and I would like to extend the model with single cell data. What is your take on integrating multi-omics with VAE for phenotype prediction?
Yeah, I've seen papers that tackle this where they have different modalities come in, and those are captured with different parts of the neural network. So you can have one neural network for like the transcriptomics data, another part for the protein data. You can have another part for the imaging data, and then you bring them all into the same latent space. So you have all these different kind of encoders, but they all converge on the same latent space. That's one technique that I've seen for doing that.
Right. And I guess the last question in the chat is, during the experiments, are the cells sampled from the same source? In other words, initially have some kind of grid of cells? If so, Have you considered having some graph file architecture for simplicity as CNN?
Sorry, can you say that one more time? I did not catch all of that.
During the experiments, are the cells sampled from the same source? In other words, do you initially have some kind of grid of cells? If so, have you considered using some graph file architecture for simplicity as CNN?
I'm really sorry. I didn't catch the first part.
Can you see? It is actually in the chat.
Yeah, let me see. Let me open up the chat here. Where is it? Okay, during experiments are the cells sampled from the same source. Okay, I see. Sorry about that. In other words, you initially have the same kind of grid of cells. If so, have you considered some graph-wise architecture for simplicity of CNN? So the cells can be sampled from the same source, but the models are like, I mean, sometimes no. Sometimes they come from very different sources. Like if you're collecting data from different studies, or you want to integrate data that comes from a different study with the data that you generated in your lab, then they will not have come from the same source. So you do not get the same. You do not. Yeah, those cells can have very different properties depending on where they came from, and in other words, you initially have some kind of grid of cells. So the cells are not in a grid. So there's not a spatial orientation to these cells in single cell RNA sequencing. The cells are disassociated from the tissue. So they're free floating. And so all you get is a set of vectors, and they don't have a relationship to one another. There is another kind of data called spatial transcriptomics or spatial genomics, where you maintain the spatial location of the cells that you're measuring. And in that case, yes, I have seen graph neural networks employed for that kind of data, where you use a graph of cells of where you have an edge between two cells if they're neighbors within the tissue. But in the data that I talked about today, there is not that spatial relationship between the cells that's maintained in the experiment.
I guess we don't have any other question. We can end the talk here. Thank you so much, everyone. And thank you so much, Dr. Matthew, for your time and insightful session.
Thank you so much. It was a pleasure speaking with you all.
Take care, everyone. Bye.
Bye-bye.